This is Chapter 16, Non-Specific Defenses of the Host. So it's not a very long PowerPoint. What it's trying to get at is that 24/7, you have ways in which you either can stop a pathogen from entering, or if it does enter, to address it immediately. And if those non-specific defenses are able to either stop the pathogen from getting in the body or kill it as soon as it gets into the body, then there is no disease. So these are some definitions. Susceptibility and resistance. Some people are very susceptible to anything that comes along, and other people seem never to get sick. So very often when that happens, people tend to talk about genetics. They talk about the ability of these non-specific mechanisms to be more robust in other people. And those are the ones that are resistant to be less robust in susceptible people. Non-specific versus specific. This chapter is all about non-specific, meaning it doesn't matter. It doesn't matter if it's a fungus, it doesn't matter if it's a bacteria, it doesn't matter if it's a virus. All of these pathogens will be treated in the same way. On the other hand, specific resistance kicks into place when the non-specific fails. It could be too many pathogens, it could be the person is stressed out and then their immune system is not robust because of that. And then when you get a specific mechanism, which will be the next PowerPoint, then you actually can develop memory. You don't do that with non-specific, but if you do have to go to specific, it's because your body is not responding and getting rid of the pathogen's getting the upper hand. So specific resistance is a way of knocking that down. The person feels sick, the person has a disease, and then the specific resistance will kick in afterwards. And if everything is fine, then it will be able to totally eliminate that invader and give you memory. Okay, so these are some of the non-specific 24/7 mechanisms everybody has. The first line of defense, keep them out. Intact skin because it's dead keratinized layer. Mucous membranes with their secretions. Your upper respiratory tract is what I'm going to use as an example of that. And it has the ability to make mucus. Now, the mucus is not the thick mucus that we think about when we're sick. It's a very watery mucus, but it can trap dust particles, which can be how pathogens enter your respiratory tract. And then the pathogens on the dust will, thanks to gravity, be trapped in the mucus. And then the cilia from the cells will create this escalator, and it will then just push the pathogens up into the mouth where you sneeze them out, you cough them out, you swallow, and then of course hydrochloric acid in the stomach will get rid of them if you do swallow them. And then of course you have your normal microbiota, and that happens, that helps a lot in your large intestine. So food poisoning bacteria really want to get to the large intestine to grab hold of the surface of the cells of the large intestine, colonize, make toxins, and make you quite sick. But if you have a good normal flora colonizing every inch of your large intestine, then it's very difficult for the pathogen to gain a foothold. So that is another non-specific mechanism. Second line of defense is if they get in. So phagocytosis, we'll get into in a second. Inflammation, fever, and at times there can be some antimicrobial substances that are made by inflammation. Then the specific is going to be the next chapter, and that's going to be lymphocytes, but we'll cover them at that time. All right, so physical and chemical factors. Keep them out. Skin, outer dead layer of keratinized cells. Keratin is great waterproofing. It acts as a barrier. The mucous membranes was ciliary escalator. So microbes that are trapped in the mucus, transported away from the lung to be coughed out or swallowed, and then the hydrochloric acid in the stomach, if they're swallowed, will kill them. Okay, normal. We have normal flora on our skin. We have normal flora in our large intestine, etc. And especially the large intestine is going to be competing with pathogen. And since they're there first, then unless pathogen has some tricks up their sleeve, then that will prevent food poisoning, diarrhea, dysentery from happening. All right, so we're going to get into inflammation and phagocytosis because that's a 24/7 if they get in type of situation. And it can be very quickly nipped in the bud. So the person isn't even aware that they've been exposed because the pathogen is going to be eliminated before it can cause disease. The most important white blood cell is going to be the neutrophil. And the next chapter, I'll get into lymphocytes. I'm not really going to get into basophils, eosinophils, or monocytes. If you notice, they're quite low as far as percentage. The majority of your circulating white blood cells are phagocytic neutrophils. And these white blood cells are specifically there 24/7 to guard you against any microbial pathogenic invasion. There we go. Phagocytosis comes from the Greek, actually, which means phago, to eat, and cytosis, to eat cells. So what it's able to do, this phagocytic neutrophil is very amoeboid-like, so it can change shape. And therefore, it can come across a pathogen, and it has this generalized sense of self, you, and non-self pathogens, and it then can recognize the pathogen and engulf, internalize it, and then kill it. So ingestion of microbes, or let's just say ingestion of microbes, are performed by phagocytosis. Later on, I'll get into particles by a cell, but right now, let's get rid of the pathogenic microbe. You can do it by phagocytosis. All right, so this is a generalized series of events that a phagocyte is going to do when it encounters a pathogen. So here's my little yellow phagocyte, and here is the pathogen. Oops, sorry, I've got a little ahead of myself. Getting there. Sorry. There we go. I promised not to touch anything. So for number one, you have the microbe. Pathogen is going to be recognized as foreign or different by the phagocyte. It is then going to be ingested. It's going to be engulfed by the phagocyte because it's so amoeboid. And then in number three, you're going to form a vacuole. And we call that vacuole a phagosome because that's what it engulfs. This is the pathogen. Now, you want to kill it. You want to poison it. You want to degrade it. And that's the job of the lysosome. So the lysosome in number four has poisons inside of it, and it has digestive degradative enzymes inside of it. So it's going to fuse with the phagosome. And then in number five, that's going to kill it. The degradative enzymes try to degrade it and could recycle some of the parts, but there's always going to be some residual undigested bits. So that's the number six. And that we call it a residual body because it has residue in it. And then in number seven, you are able to bring it to the cell membrane and do exocytosis. It will exit the cell, and that's the end. That's how phagocytic neutrophils are able to kill pathogenic microbes. Now, inflammation. The classic signs of inflammation is redness, pain, heat, and swelling, which is called edema. The big thing and the really major inflammatory modulator is histamine. It's going to be released by mast cells, and I'll get into them a little bit later when we get into the next chapter. But these cells are scattered throughout your body. If indeed there is a trigger, usually entry of pathogens or injured cells, you are going to have chemical signals tell the mast cells to release histamine. It will also release other types of molecules, but histamine is really the key for inflammation because it can bind to all kinds of cells in that general area where it's infected and can promote increased capillary permeability, increased vasodilation, and smooth muscle contraction. Now, when I say histamine is responsible for the 4 cardinal signs, let's bring it into the increased capillary permeability. The whole idea of histamine binding to capillaries is to pull them apart slightly so that the phagocytic neutrophil can kind of squeeze its way through to the infected tissue. And there, it will be able to engulf the pathogen and kill it. You're not just going to have the phagocyte squeeze its way through. Plasma, 70% water, is also going to be able to move from the capillary into the wound area, and that's what's going to cause swelling. So the edema or the swelling is caused by that. But when you have an area that is starting to swell, pain receptors, you have receptors that are sensitive to pressure. They're going to now fire. And the upshot of that is our perception of pain. Now, the other thing that happens with histamine is that it can bind to small arterioles and cause them to dilate. You want that because you want to have the delivery of the phagocytes to be very rapid, and that can be enhanced by vasodilation. However, when you dilate, especially if it's an area near the skin, that skin area is going to be red. Why? Because most of the blood cells are red blood cells and they're red. So when you vasodilate, that area is going to appear redder. And the other thing is heat. When you dilate, you increase the turbulent blood flow, and that will create heat. So the heat and the redness is caused by the vasodilation of the arterioles feeding the wound area or feeding the infected area, and then the pain, the swelling, is caused by the binding of the histamine to capillaries. I just wanted to mention smooth muscle contraction. It is not good in the respiratory area because your bronchi and bronchioles align with smooth muscles, so when they contract, it gets very difficult to breathe. But in every other place of the body, that's a good thing. When your smooth muscle contracts, and every organ system has a layer of smooth muscle, and it becomes quite rigid, and that is going to prevent the spread of the microbe. And sometimes you can feel it as cramping, but on the other hand, about the only place that it becomes perhaps dangerous, very dangerous, is when you start to bronchoconstrict and close the air passages. Okay, so this is just showing you what I was talking about, and I just want to go back one. I don't know why this thing is jumping. Come on. Yep, okay, so that is the next one. So this is just showing you a splinter. So, you get a splinter, there are bacteria on the splinter, and then you have the release of histamine. Now, these are other types of signal molecules that are involved in inflammation, but the kingpin of inflammation is histamine, and that's the only one that's going to be on the quiz for inflammation. So then, obviously, the splinter, you're going to have blood clotting, so you don't bleed to death. But then that whole cascade is going to start to trigger the mast cells to release histamine, and that whole area is going to get swollen, painful, hot, etc. And that's due to the histamine binding. All right, next slide. So this is kind of up close and personal. When the histamine binds to these are actually cells. You don't really, well, you sort of see them. These are called endothelial cells. Binds to it, and then they pull apart just a wee bit. So your phagocytic neutrophils can kind of squeeze out into the area that the pathogen is in. And that's fantastic. In that, if it engulfs and kills them, then you have closed the wound, you have removed the pathogens and any injured cells, and now tissue repair, wound healing can occur. All right, fever. Fever is something that is not generally invoked all the time. But through the higher scale of evolution, the body has recognized that certain pathogens are more dangerous. And fever is another tool by which you can kill and aid in the process of inflammation. It's another way of killing. Um, I'm going to conjecture that the reason why fever is not standard for everything is, the risk that you always take that it's going to, especially in the very young and the very old, overshoot. And there's a marked difference between, 103 and it overshoots to 108. And at 108, it could kill you, and 103 is effective. So with the very pathogenic organisms, think about the flu, influenza virus. It killed more people in 1918 than every soldier who died in World War I. So that is considered to be a very dangerous pathogen as far as the body goes, and fever is invoked. So how do you do that? Well, the body temperature is regulated by the hypothalamus, and that's at the base of your skull, and it's communicates, with the pituitary gland, etc. But that center, hypothalamus is a thirst center, it's the temperature center, it's a lot of different things. But right now, let's talk about how you get fever. So the fever cascade starts with the pathogen. Now, they're going to use gram-negative endotoxin, and that's just one of many, many triggers. So with the influenza virus, it's probably something on the outer coat of the virus that is going to trigger. When a pathogen triggers a fever response, whatever chemical they are going to present to the immune system, it is called an exogenous pyrogen. Exo meaning it came from the outside, and pyrogen meaning that it's going to cause a fever. But the pathogen causes the fever. That's how the body can distinguish between a pathogen that's not going to cause a fever and a pathogen that does. And all of this was established millions and millions of years ago. So when you have that exogenous pyrogen in there, then the phagocytes are going to release something quite different. And it's called an endogenous pyrogen. There should be a little thing there. And it's going to release a signal called interleukin 1. Very specific signal. And that is going to result in the making of prostaglandins. Another signal, but this signal is very specific. Prostaglandins interact with the hypothalamus to reset that temperature at a higher level. So the way that the cascade goes is the pathogen is actually going to start the ball rolling. It becomes the exogenous pyrogen. Then that triggers a phagocyte to make something that it normally doesn't, which is interleukin 1. Interleukin 1 is responsible for the generation of prostaglandins that reset the hypothalamus to fever, like 101, 103. The fever is a carefully balanced higher temperature to be very bactericidal, virucidal, fungicidal. but not so high that it damages your own cells. And then once that has been, meaning that pathogen has been eliminated, then you don't have the, and then then the phagocytes will stop making interleukin-1, which will mean that you're not making prostaglandins, and the hypothalamus will reset back to normal. And that's it. So I will see you for the next chapter, which is going to be the specific lymphocytic immune response with memory. See you then.